The last class, we talked about an explanation for the red edge. The red edge is this line here, which goes almost straight down at 700 nanometers, at which point the leaves on the plants start reflecting instead of absorbing the light. At shorter than 700 nanometers, all the way down to about 210 or 220 nanometers, they absorb very strongly, about 95%, except for a little dip here, which is at the green, about 550 nanometers. And that's why we see when we walk in the forest that the trees and the plants are green. It's also because our eyes are a little bit more sensitive in the green. But the interesting question is, why do we have this red edge? Why does it exist? Why do plants and trees all of a sudden stop absorbing at about 700 nanometers and start reflecting, and very strongly reflecting, all the way down to about 1.2 microns, where water starts absorbing again? But there's a range between 700 and about 1.2 microns in which the plants are reflecting. Now, the biological explanation for this, or the reason given for this by the biologists, is that it has to do with the electronic structure separated from the vibrational structure of the molecule by a certain energy gap. And they claim that this is in the energy gap. So below about 700 nanometers, we have electronic absorption. So the electrons are going to a new excited state. And above 700 nanometers, well, really about 1.2 microns, then we get the vibrational states, so the absorption through vibration. That is the traditional explanation of the red edge. But in fact, so what they're suggesting is that there are no molecules which have states, energy states, within the range between 700 and 1.2 microns, 700 nanometers and 1.2 microns. But in fact, we know that there are states in that region, and we'll show that in a minute. But we have for the plants, for example, this is now the reflection. So instead of absorption going down at 700 nanometers, the reflection goes up. So for plants, mosses, algae, and aquatic plants, all the plants which are at the surface of Earth, they reflect very strongly at 700 nanometers. But once you get down to the lichens and the bacteria, which are at the ground level under the canopy of the forest, then the red edge does not become very obvious. No, it starts to disappear. And when we get to the bottom of the oceans, for example, the bacteria at the bottom of the oceans, there is no red edge, or not very definable red edge. So that gives us an indication that it has to do with the intensity of the light, because those which have strong reflection at the red edge are those which are exposed to the greatest intensity of light. And those which are at the bottom of the canopy of the forest or the bottom of the oceans don't have a well-defined red edge. Okay, so I gave some indications that it's not really optimizing photosynthesis, which is behind evolution in nature. So for example, common plants and trees have a photosynthetic efficiency of about 0.1 to maximum 2%. So that means only 0.1 or to 2% of the free energy which is absorbed on the leaves of the plants are actually used for fixation of carbon through photosynthesis. So that's very low photosynthetic efficiency. So if nature was optimizing the reproduction, for example, the growth of plants, the spread of plants, then you would expect the photosynthetic efficiency to be much higher than it actually is. And just an example, for example, humans have made photovoltaic cells which have efficiency of about 20 to 40% of converting sunlight, the free energy in sunlight, into electrical energy. So even though nature has had many thousands of millions of years to evolve higher efficiency, photosynthetic efficiency, it's remained relatively constant, even lately, of between 0.1 and 2%. We also see that we have a saturation, that once we get over a certain flow of intensity, which corresponds to about 10% of the energy of the sun directly overhead, then we get to a maximum in photosynthetic efficiency. So it's obvious that at least 90% of the light which is arriving at the leaves of the plants are not being used for photosynthesis. Okay, I'm going to skip over most of what we took yesterday or last class, just to refresh your memory that the absorption spectrum is very strong, about 95% over the whole range up to 700 nanometers when we have this red edge, and then it starts absorbing again about 1.2 or 1.5 microns. And that's basically due to the absorption of water in the infrared. And so then we discussed the different pigments which are in the leaves of the plants. One of the pigments is RNA and DNA. And these absorb in the UVC range, or the UV lejano range. Around 260 nanometers, they have their maximum. And amino acids also absorbing very strongly, about 260 to 280 nanometers, especially the aromatic amino acids, which have this aromatic ring. And the mycosporins, which are absorbing in the near UV, at about 320 nanometers. And we also have carotenoids, which absorb in the blue, and now in the visible. And of course, we have the porphyrins, which are the chlorophyll molecules, which are absorbing in the blue and also in the red. We also have flavonoids, which absorb in the blue-green region, about 500 to 550 nanometers. And we have phycobilins, which absorb in the yellow and the red. So all of these together, they provide this very strong absorption until the red edge at about 700 nanometers. But you can see in this spectrum, this is the spectrum of the sunlight arriving at Earth's surface today. You see that 700 nanometers, we're missing about 51% of the sunlight. 51% of the sunlight is not being used by the plants. It's not being absorbed, it's being reflected. So that's a lot of free energy which is being just reflected. So why would plants be doing that, especially when the photosynthetic efficiency is maximum around 700 nanometers? So that means that for photosynthesis to be optimal, plants should be absorbing around 700 nanometers. So as I mentioned, the explanation from the biologists of the red edge is that there is a space or a gap in energy between the vibrational states of the ground state and the first excited electronic state. And that's why that beyond 700 nanometers, there are no pigments which are absorbing. And they claim that this has to do with natural chemical physical reasons of the molecules, the pigments. But in fact, we know that that is not the case. As I mentioned, those bacteria which live at the bottom of the ocean, where the intensity of sunlight is 0 or very small, they in fact do absorb just in this region here. And this is a spectrum of the absorption of chlorophyll, but bacterial chlorophyll. So we see that there is absorption for some molecules, some pigments within this red edge. But why don't we find that for the plants which are exposed to higher intensity sunlight? So therefore, the reason given by the biologists for the gap here, about 700 to 1.2 microns, is not consistent with the data, because in fact we do have molecules which absorb in that region. And these are the chlorophyll B molecules and the bacterial chlorophyll A and B. And just to give you some examples, these are bacteria that absorb within this region in which the plants and bacteria on the land are not absorbing. There are in fact bacteria which live under low intensity light, which are absorbing in this region. So then one would expect, if nature was optimizing photosynthesis, then we would have maximum absorption at about 680 nanometers, because that is where photosynthesis is optimized. If you do experiments in the laboratory with a light source which can be changed, its wavelength or its frequency, then we find that we have maximum photosynthetic efficiency at about 680 nanometers. So that corresponds here to this peak. I don't know if you can see my pointer. It seems to disappear every once in a while. It has to do with the slow rate of my computer, but it's the first graph on the upper row towards the left. So that is what we would expect the absorption to be like if we expected that nature was optimizing photosynthesis. And this is the spectrum of light which is arriving at the Earth's surface, this inner line here. The outer line corresponds to the spectrum at the top of the atmosphere. So there's some absorption in the atmosphere, and these little spikes here are due to the absorption of water in the atmosphere. So if we convolute this absorption spectrum that we would expect if photosynthesis was being optimized with the light spectrum that we do get at the surface of Earth, then we should find something like this. So this is just a convolution of these two graphs. We should get spectrum that looks something like this. But in fact, we get a spectrum which looks like this. So they are quite different. So when we convolute the actual absorption spectrum that we measure in the leaves of plants with the spectrum of sunlight arriving at Earth's surface, that gives us this graph. So how can we explain that? So let's try to explain it by assuming that plants are not optimizing photosynthesis, but are optimizing entropy production. So therefore, if there was a limit to the number of photons that the pigments could handle, because of their finite size and because of their finite lifetime, remember that if you excite a pigment, it can no longer absorb another photon until it de-excites again. So there's a limit both to finite size and to finite lifetime. So if nature was optimizing entropy production, or the dissipation of photons to produce entropy, instead of optimizing photosynthetic efficiency, then it would like to absorb those photons of shorter wavelength. That's because when you dissipate a shorter wavelength photon into the infrared, you produce more entropy than if you dissipate a longer wave photon into the infrared. So we can do the calculation of the entropy production. It's just the outgoing flow of entropy, which is due to the emission after absorption of the photons in the plant leaf, and the part which is due to reflection and transmission minus the incident entropy of the photon beam, which is incident, which is given by this spectrum. And therefore, we can calculate the entropy production as a function of number of photons starting from this short wavelength, because at the short wavelength region is where we get more entropy production per photon. And if it's a question of number of photons that the system can handle, because of its finite size and its finite lifetime, the system will start to dissipate photons which are at the short wavelength region because they produce more entropy. So this was just an example of the different sizes of the pigments. And we can make an estimate that given these finite sizes of the pigments, we could have a maximum of about 2.6 times 10 to the 16 pigments per meter squared. And that is roughly what they calculate from satellite photos of photosynthetic areas over forests, for example. And if we know the lifetimes of these pigments, and we assume that we have an average lifetime of about 0.5 nanoseconds, then we can easily calculate that we could, calculate about 5.2 times 10 to the 23, I can't read it, but photons per second per meter squared. I can't read it because there's a silly bar which I cannot get rid of here on the screen. So I hope I got that number right. And so let's try to do the calculation of the entropy production, simulating the spectrum. So this is the actual measured spectrum at Earth's surface. And this is what we were able to simulate. Okay, it's just a black body spectrum, about 5,700 degrees centigrade with some absorption for water, basically the absorption of water for these lines here. And let's do the calculation. So this is the spectrum that we get for the absorption. It's a function of wavelength of the leads. And so let's start at the short wavelength side, count the number of photons, and then get to this number of 5 times 10 to the 25. So this is the number that I should have said on the last slide, 5 times 10 to the 25 photons per meter squared per second. So that's what pigments in plants today can handle, given their finite size and given their finite lifetimes. And so we just start counting the number of photons in each wavelength bin until we get to this number of 5 times 10 to the 25. And when we do that, we end up right here, almost perfectly where it's found in nature. So this is the simulation, assuming that we are optimizing entropy production rather than photosynthetic efficiency. And we get almost the same absorption spectrum. Of course, within the limits of the approximation of the model to the reality, we get very, very close to the red edge, about 700 nanometers. So the red edge then is just due to the fact that, well, according to our theory, that plant leaves cannot absorb and dissipate more than about 5 times 10 to the 25 photons per meter squared per second. Because beyond this, you just saturate the system. And so those photons are going to be reflected or transmitted because the pigments are going to be in their excited state and will no longer be able to absorb. Okay, so that's the theory, and that's the reason for the red edge, according to our theory, which is based on maximization of entropy production. So now we can make some predictions. So one of the predictions is that The red edge is going to depend on the environmental conditions, in particular, the intensity of the light. Because the more photons there are coming on the leaf, the less towards the red edge they're going to be absorbing because of their finite limits. So for example, if we have leaves which are below the canopy or below water, where the light intensity is low, then the red edge will move towards the red. Because we have low intensity light over the visible range, so we still can dissipate photons, so we're still going to absorb more photons towards the infrared, more towards the infrared. And this is confirmed in the absorption spectrum of bacteria and lichens. Now, if we go to high latitude, where the sun is lower in the sky, we have less intensity light, and so the red edge should be moved towards the red also. We go to high altitude, we have a higher intensity of light because we have less absorption in the atmosphere, so the red edge would move towards the blue. So these last two predictions can be verified. So we have really a way to check our theory by comparing the red edge of plants at high latitude and high altitude and looking to see exactly where the red edge falls. Now, if we also have plants with a higher density of pigments, so remember we assumed that we had a finite size of these pigments and that we had a finite area or a finite density within a given area or areal density of pigments due to their finite size and due to the fact that we need water, we need some kind of support structure. So for example, in these chloroplasts, we have these thyloids or thicholoids, I think they're called, these little black spots in which all the organic pigments are located. And around them we have water because we need to dissipate. If we don't dissipate the energy, the electronic excitation energy, then as I said, we cannot absorb another photon. So that gives us a limit to the number of pigments per unit area. But some plants have evolved such that their density is higher. So if we have a higher density of pigments, then we could absorb still more photons towards the infrared. And in fact, this is being confirmed in some of these plants, which have chlorophyll at a higher density by these people. So in that case of high density pigments, then we can move further towards the infrared. So this prediction is confirmed. We can also have a prediction that plants which transpire a lot can reduce their dead time. So if there's a lot of transpiration, what you're doing is taking any energy away rapidly from the leaf. So the energy, of course, is the energy of the photons. So they're de-exciting much more rapidly. And it turns out that the old form or the more ancient form of photosynthesis, which is known as C3, which transpires more than C4, has also the red edge moved towards the red. So this is like another confirmation data which is already being determined. Okay, so are there any questions about the theory? Now I'm going to get into a little bit about detecting life or suggestions for detecting life on other planets of other stars. If you have any questions about what I just went through, please stop me. Okay. So now the red edge is something quite well defined, especially if you're very close to the vegetation. So as we saw in this, well, let me find a better graph here. In this graph, the red edge is very well defined. So the reflection here is very large. So that should show up. If there are planets on other planets of other stars, extra solar system stars, then maybe we can detect the red edge. Okay. But be careful, because biologists think this is something inherent in nature, that this red edge would always be the same at 700 nanometers. But we have just shown that it depends on the intensity of the light. Depends on the intensity of the spectrum of light arriving at the surface of the planet. So it could move towards the red or towards the blue, depending on the intensity of the light. But anyway, they are going ahead with this program to see if the red edge can be detected on planets of other stars. So to test this proposal, what they decided to do first was to see if that we could detect the red edge on Earth from the light scattered off the moon, the dark side of the moon. So here in this diagram, at the bottom left, we have the sunlight coming into the Earth, being reflected by the dark side of the moon. And then we detect the light that is reflected in this region of the dark side of the moon, which is the light which is being reflected from Earth. So if we can detect the red edge in this light, which is being scattered off the moon, first scattered off the Earth and then scattered off the moon and then back to Earth, if we can detect the red edge in this region, then possibly we could detect the red edge on planets of other stars. So that was the first test that they did, and here are the data. So this is for different orientations with respect to the dark side of the moon. This is the case with South America here in view of the moon. And this is the case of purely ocean view of the moon. So we look at about 700 nanometers here. And we don't really find a red edge. Maybe there's a little bit of the red edge. Some people thinking that it may have been shifted towards the infrared because of the other processes which are involved, like the water cycle, for instance. So the water cycle can absorb, or the water in the atmosphere can absorb some of the light which is reflected beyond the red edge, and then shift that light still further towards the infrared. So the conclusion is that it's going to be very difficult. We can barely detect it on Earth, the light which is reflected of the moon, maybe here or maybe here. But it can be barely detected, and only perhaps over land areas and not over ocean areas. So it's going to be difficult to detect anyway. And the further problem is that, as we have mentioned, that the position of the red edge, whether it's at 700 nanometers, at 600 nanometers, or 800 nanometers, etc., would depend on the intensity of the light arriving at the planet, at the surface of the planet. So we cannot expect to find it at 700 nanometers. And now we see that we also have this extra problem of coupling of different irreversible processes, which could shift the reflected light further into the infrared, further dissipation of the infrared light. So it smears out the red edge, so we don't see the red edge. So then, just to recap, to summarize, the biologists think that the pigments which are in the leaves, which are absorbing from about 210 to 210 to 700 nanometers, they suggest that these pigments are for photoprotection. So for example, in 2001, this biologist wrote that the regulated thermal dissipation of absorbed light is without question the keystone of photoprotection. So they are saying that these pigments in the absorption are for photoprotection. But we know that these molecules don't have to absorb. They could reflect or they could transmit. And you can easily change the structure so that they don't absorb in the regions, which could cause damage to the photosynthetic system. So photoprotection is only to protect the photosynthetic system. And in fact, when they actually try to do the calculations of which wavelengths could affect the photosynthetic system, well, they in fact find, like, for example, the absorption of chlorophyll is only at 430 nanometers in the blue and about 680 nanometers in the red. And between that, there's very little absorption, so that light is not going to affect at least the chlorophyll pigments, which are used for photosynthesis. So what I propose then is that the regulated thermal dissipation of absorbed light is without question the keystone of evolution. So that is in fact what nature is after. It's after the dissipation of photons. So that's what it's trying to optimize. And it's a complex system right now of the biosphere, which is very complex, and the different hierarchical levels which are nested. But the basis of the dissipation is the absorption and dissipation in the biological pigments which are on the surface of Earth. And so that is what nature is optimizing, is to dissipate photons. Okay, are there any questions or comments?
Yes, I have a question.
Yes.
The red edge has been modified through the stage of art?
I'm sorry, Ricardo, is it Ricardo? I can't see your name. Yes,
Yes.
Ricardo.
My question is, the red edge has been modified through the stage of art?
Sorry, I didn't understand H.
Stage. Through the time of Earth.
Yes, through evolution.
Yes. Has been modified.
Yes, I suspect so. That's in fact one thing that we're looking at. We have one paper published which looks at primitive pigments. So there's some pigments which you can even consider are fundamental molecules that they are in the three domains of life. And we have been looking at these pigments, these fundamental pigments, and these pigments, in fact, are absorbing prominently or basically in the ultraviolet and in the UVC range. So that gives us an indication that the earliest pigments arose to dissipate the shortest wavelength light, which was arriving at the surface. And then over evolutionary time, gradually more and more pigments were developed or produced or synthesized to absorb light more and more towards the visible. And now it goes all the way to the red edge and even a little bit beyond in some of the bacteria which are in very low light regions on Earth's surface, or below the ocean or below the canopy on Earth's surface. So yes, there seems to be, we have to do that in more detail, but we have an article which if you remind me or send me an email, I'll send to you, about ancient pigments and how we're finding that they are correlated with their absorption spectrum to the spectrum which was arriving at the surface. So it would be natural, as I mentioned, for nature to dissipate the shortest wavelength photons because they produce the most entropy when they are dissipated. So yes, Ricardo, there appears to be an evolution of the pigments from the ultraviolet towards the infrared. So maybe in another 1,000 million years, we will have more and more pigments which are absorbing more towards the infrared.